All right, well, I'd like to welcome everyone to today's edition here of the Polyploidy Webinar. This is our first meeting of the 2024-2025 season. It's great to have a couple of folks here joining us who, well, one of these first here is Shelley Gaynor. And Shelley, I think we all know Shelley is recently graduated with her PhD in the Soltis Lab back in August and has currently started as an NSF postdoctoral research fellow in biology, one of the PRVs, working at the University of Michigan with Stephen Smith. Shelley, I think many of us in the polyploid community know Shelley and her great work. She's spoken in the past, spoke here in the past back in I think 2020 maybe or 2021 to us in the polyploid webinar about or maybe in earlier versions of some of these models. She's here today to talk to us again about these great work that she's been doing on computationally intensive methods and models to understand mixed cytotype populations. With that, I'll let Shelley take it away.
Thanks, Mike. So today I'll be talking about AutoPop and NQuack, which are two softwares that I developed during my dissertation in collaboration with Jacob Landis, Nick Cortezis, Tim O'Connor, Rob Laporte, Jeff Doyle, Pam and Doug Soltis, and Jose Miguel Ponciano. And even though this is the polyploid webinar, I wanted to start with some basics. An autopolyploid is when we have whole genome duplication within a taxon. So this can happen through unreduced gametes from a single diploid individual or from two closely related individuals. Those can come together and make our purple tetraploid. Alternatively, a blue unreduced diploid gamete can come together with a reduced diploid gamete, create your orange triploid, and that orange triploid plus a reduced diploid gamete can come together and make your purple tetraploid. I only stress these colors because they're consistent on the slides, but they don't have any biological meaning. Once your autopolyploid forms, it faces positive frequency-dependent selection, and it has no one to mate with. So it's the minority cytotype, and it's being excluded. And so to escape minority cytotype exclusion, theoretical studies have suggested a couple different mechanisms. First, you could have spatial segregation, so your tetraploid can move into a new geographic habitat. Alternatively, you could have phenotypic divergence, so shifts in flowering time per se, and both of these are thought to be mechanisms of reproductive separation. Another way to escape minority cytotype exclusion is through reproductive assurance, through asexual reproduction. And lastly, recurrent formation is thought to assist in escape from minority cytotype exclusion. In the system that I focus on, Galix uceolata, a southern Appalachian endemic with diploid, triploids, and autotetraploids, it really doesn't have a lot of things going for it. It lacks that reproductive isolation. There's geographic overlap. There's no phenotypic divergence. Gene flow is quite likely based on microsatellite markers and target enrichment, as well as because of their overlapping flowering time and shared pollinators. It also has no reproductive assurance. It's clonal, but it's self-incompatible. So the only thing it has going for it is this recurrent formation. Despite this, when you walk into the populations, you see a mishmash of cytotypes, and there's no microgeographic segregation. And this really has motivated the softwares and the methods that I'll show you today, because I wanted to know, could these actually persist, or was this only a snapshot? And I also did a lot of flow cytometry to create the graph you see here, so I wanted to create a tool to not have to do flow cytometry. So today I'll first be talking about AutoPop, which is asking that question if these mixed cytotype populations are only snapshots. And then I'll talk about NQuack, where I try to leverage sequence data to predict ploidal level. So starting with AutoPop, this paper was tentatively accepted, so hopefully it should be out in the next couple months. And the goal here was to investigate the formation, establishment, and persistence of diploid, triploids, and autotetraploids over time. I wanted to know the probability that diploids and tetraploids would coexist and the stability of that coexistence. So in populations of Galix, would this be something that is only that snapshot, or could this persist over time? And so to investigate this, I built my own population simulations based on a stage-structured matrix population model. This includes demographic stochasticity, so variance in the birth-death process, and environmental stochasticity, or variance in population abundance. And instead of getting into the nitty-gritty mathematical details, I'm going to go through a diagram and show you what happens in a single time step. So first, our model includes reproductively immature and reproductively mature individuals of each of these three cytotypes. At each time point, we first have survival. Here we have immature survival dictated by environmental stochasticity as well as demographic stochasticity. So there's a density-dependent process here. For mature individuals, survival is only dictated by environmental stochasticity. At each time point, immature individuals can also become mature, and then we can have more individuals in our population through reproduction, which is also a density-dependent function and includes selfing and mating proportion. And since this is a cytotype-specific mechanistic function, I'm going to walk through it a little more. So first, in this reproductive step, we have gamete formation. So here, diploids can form unreduced or reduced gametes. Triploids can only have viable 3N gametes, and tetraploids can only have viable 2N gametes. And the number of gametes produced by each individual is density dependent. Of the gametes formed, a proportion of them can go on to outcross, but based on a selfing rate, a proportion will go on to self. Those that outcross can either have group-based assortative mating based on a mating proportion, so this means that you can only pair with gametes that were created from like cytotypes, or they can randomly mix. And here you might notice we have unviable offspring, and this is anything above a tetraploid. What's really important to highlight with our reproductive function is the fact that diploids can only be produced by diploid gametes, but triploids can be produced by diploid or autotetraploid gametes, and autotetraploids can be produced by diploid, triploid, or autotetraploid gametes, and this is unique to our model. So just to summarize, our model is looking at perennial, long-lived flowering plants with overlapping generations. We have incomplete reproductive isolation among the cytotypes, and we always set equal parameters for our diploids and our tetraploids because we want them to have equal competitive ability. Because I wanted to simulate formation, we start with a diploid-only founding population, and we also allow recurrent formation. With this model, I investigated 4,356 parameter sets, and these focused first on reproductive isolation, so that's selfing and mating proportion. Next, we look at variation in the environment, so low, medium, and high, but we also incorporated a correlation in the response to the environment among the cytotypes. So cytotypes were able to respond to the environment differently, so have niche differentiation, or they could respond to the same environmental conditions in the same way, so niche similarity. Lastly, we looked at different types of cytotype interactions. So I mentioned that we have density-dependent immature survival and gamete production. Here, this could be based on only like cytotypes or all individuals regardless of cytotype. So for each of these parameter sets, we did 500 replicates for a little over 2 million simulations. And since that's a whole heck of a lot of simulations, I'm going to focus just on that high cytotype interaction. So when density dependence is based on all individuals regardless of cytotype. So what did we find? Here, you're looking at the probability of extinction and fixation as a function of mean abundance. And we see that as mean abundance increases, the probability of fixation increases for our diploids. For our tetraploids, we see that the probability of extinction decreases, however, with increased mean abundance. And this fits with what other studies have found, that an autotetraploid must outcompete its diploid progenitor to persist. However, we also looked at coexistence, and we did find that mixed cytotypes can persist in symmetry. And we have this peak here where selfing rate is high. However, it's not as clear, but when you increase your environmental variance, here we're looking at low environmental variance. When we increase to medium, we can see that relationship between selfing rate and these values. So as you decrease your selfing rate, that probability of fixation increases for your diploid. But as you increase your selfing rate, that probability of extinction decreases for your tetraploid. And again, that peak here for coexistence, you have a higher probability of coexisting when your selfing rate is high. When you increase the environmental variance even more, you lose this pattern. However, I do want to highlight that we see an increased probability of extinction for our diploid, and we see an increased probability of fixation for our tetraploid, which is quite interesting because it's been hypothesized that tetraploids will persist in times of high stress. Think of the PK boundary. And so this finding was quite interesting because we don't have genetic flexibility in our model. So to investigate this further, I looked at the intrinsic growth rate, which is the log scale change in population abundance over time. So here you're looking at the tetraploid intrinsic growth rate and the diploid intrinsic growth rate. across different selfing rates for low environmental variance. And if points fall above this line, we say it's a tetraploid advantage. If it falls below the line, you have a diploid advantage. So as you increase your selfing rate, so you go from 0 to 1, you see more points above that line. So you see this tetraploid advantage increases with increased selfing rate and increased mating proportion. When we look across environmental variance, so we increase our environmental variance, we see even more points fall above this line. So we see an even stronger tetraploid advantage when environmental variance is medium or high. So why do we see this tetraploid advantage? When all parameters are equal, when they have equal competitive ability, the only difference is reproduction. And like I mentioned earlier, diploid gametes can contribute to triploids or autotetraploids. And this is diploid gamete wasting. This also highlights that genetic flexibility isn't needed for our tetraploid advantage when you have intercytotype offsprings as a possibility. And we're not the first to bring this up. This was actually in 1975 when Donald Levin introduced minority cytotype exclusion. It was actually as a contrast to gamete wasting. So instead of thinking in terms of frequency-dependent gamete wastage, we may view this process in terms of frequency-dependent mating success. However, at that time, and as we've moved forward, we've mostly only considered these crosses. So when you have diploids creating diploids and tetraploids creating tetraploids only. When you consider all the other possibilities, you start to see the importance of gamete wastage. Additionally, with our model, we found stochastic flickering, which is a very fancy way to say the cytotypes are not in sync, and there's movement between equilibria. So this means that a frequency at a single time point only captures an instance of cytotype dynamics. And so this suggests that we probably need temporal monitoring, though I know this is tedious and impractical, and that kind of gets to our next software. But without it, we can't really fit this model to real data. So to extend this model even further and to use it to understand the history of extant mixed cytotype populations, we're including genotypes, mutation, and migration, so it can be applicable to genetic data sets. So back in 2021, when I first presented at the Polyploidy Webinar, I actually shared most of this part of the model, but now we're finally incorporating it and putting them together and using it to understand naturally occurring populations. Okay, moving on to NQuack. So NQuack is a software that was just published in Applications of Plant Science special issue Twice as Nice. Here we aim to predict the ploidy level from sequence data using site-based heterozygosity. As many of you know, there's many ways to quantify ploidy level, but as you can see by Nico asleep at the flow cytometry is very tedious. It's also tissue limited, so you have to have tissue to be able to run this. You must also have corresponding chromosome squashes to infer ploidy. And so many other approaches are used instead, including sequence-based approaches, so K-mer-based with smudge plot and tetramer, but here whole genome sequence data is preferred. Alternatively, you can do site-based heterozygosity, which is more fit for reduced representation, and this includes nQuire, Ploidy-Frost, and more, but these also recommend high coverage, and the statistical tests vary among them. With a data set that I haven't explained, but is Laria tridentata, I tried out all these methods and I ran out of tissue, and I got to a point where the accuracy was too low on our knowns that I wanted to create my own method to increase the accuracy. And so I developed a site-based heterozygosity approach. So what is site-based heterozygosity? This is an individual-based approach, so you have sequence data mapped to a reference genome, and you pull out your biallelic sites. At a site where you have an A and a C, you expect to sample an even amount of A and C, so 50% of both. And this simple idea is what site-based heterozygosity is based on. So for a diploid where you have an A, B for your heterozygote, you expect 50% of your reads to be A and 50% to be B. So you'll have this peak at 0.5. This can be easily expanded for triploids. So here we have two types of heterozygotes, an AAB and an ABB. And here you're just counting the A's and counting the B's. So we have 67% A, 33% B, 33% A, 67% B. And that's how you get your expectations for this method. So this is really easy to expand. Here we have our 3 heterozygote types for your tetraploid, your 4 for your pentaploid, and your 5 for your hexaploid. Using this basic idea of counting A's and B's, we make NQUAC. So here we process and filter your data, we estimate the likelihood of a mixture of models representing the different ploidy levels, and then we return summary statistics with predicted ploidy level and percent support. And this might look quite familiar because it's heavily based on NQUIRE. Over the next couple slides, I'll walk you through the different stages of our method and highlight what we've added, what was not included in NQUIRE. So first, data processing. Before using our method, we highly suggest that you remove repeats and reads that map to multiple regions. Once you have your beautifully cleaned sequence data mapped to your reference, our software transforms this into a table, then removes insertions and deletions, and removes sites with counts for more than 2 nucleotides. We only want the biallelic sites. Then users have options for filtering in order to remove noise and reads that aren't represented of your data, like things that are just outliers. So you can do this by filtering by minimum and maximum coverage, sequencing error rate, or with allele frequency truncations, where you pick a lower and an upper bound and truncate your allele frequencies based on these values. In the end, what you want is a table with 1 column that includes allele frequencies. We subsample the allele frequency of A or B because double sampling, which is done in NQUIRE, actually inflates signal. So it's similar to p-hacking in hypothesis testing, but here you have information criteria that is artificially inflated if you sample both. So we randomly sample one or the other. With your nice, clean, not doubly dipped data set, we then calculate the likelihood of a mixture of models. To do this, you need a distribution. So we include an implementation with a normal distribution, which spans from -infinity to infinity. But because we're using allele frequencies here, and those range from 0 to 1, why not use a probability distribution that ranges from 0 to 1, like the beta? And after thinking about this for a really long time, we also wanted to try a distribution that allows us to not convert counts to allele frequencies, because this leads to loss of original data attributes like variance and heterogeneity. So we wanted to use a distribution that mimicked the data generation process, and that's possible with the beta binomial. Like NQUIRE, we also include a uniform mixture with each of these implementations in order to allow you to capture noise. Okay, but wait, there's even more. So with a mixture model, each part of your mixture has a mean, a variance, and a proportion. You can see this here with our tetraploid, where you have your 0.25, your 0.5, your 0.75 for your 3 types of heterozygotes, but you also have an assigned proportion and variance. Now, in NQUIRE, to calculate the likelihood of a diploid, triploid, tetraploid, and so on, you calculate only the variance. You keep the mean and the proportion as a set value, as you're expected. However, we know that autopolyploids and allopolyploids have different expectations. Here you can see our duplicate alleles will have almost equal proportion among the types of heterozygotes, but for our allopolyploid, we'll have a higher abundance of 0.5. So since we know that proportion and variance of each mixture is expected to differ between an autopolyploid and an allopolyploid, and we wanted this method to be applicable to both, we also include implementation where the fixed model calculates the proportion and variance. When you're calculating the proportion and variance, the only thing you keep constant is the mean, which is the only thing we really have a good expectation for. Okay, so our method then returns summary statistics. And these first include a BIC score, which we found to lead to less error in model selection. This score is penalized by both sample size and number of parameters, but we also allow you to get a bootstrap support. So how robust is the model given the data? This is because interpretation is quite hard and math models are math and not biology. And so we wanted to allow you to resample your data and assess the reliability of model choice. Okay, to test this method, these 3 distribution types with or without a uniform mixture, plus those 3 types of fixed models, or 18 different modeling approaches, we simulated 355 samples, and then we looked at 482 real samples. These real samples included autopolyploids Galex uceolata and Laria tridentata, as well as allopolyploid glycine. In order to allow us to compare our approach to enquires, we also included their samples. So 3 samples of yeast and 2 of Oomyces. So in total, it's a little over 500,000 models. So first, I wanted to assess how enquire did on our samples. And so there's 100% accuracy for the samples included in enquire, the yeast and Oomyces. But we had quite low accuracy for glycine, only 53%, and okay accuracy for Galix at 91% and 70% for Laria. For our implementation of enquire, where we have a normal distribution with variance free and a uniform mixture, but we're not double dipping and we change our summary statistic, we are able to increase the accuracy for Galix and for Laria. However, we did not increase the accuracy for glycine. When we use the complete and NQUAC modeling set, we increase this accuracy dramatically. We go from 53% for glycine to 94%. We're also able to increase Galix and Laria even more. And so overall, we found that NQUAC is the best available software for predicting ploidy level based on site-based heterozygosity. Of course, the 78% here is not what I wanted. I wanted 100% for all of these. So I'm going to summarize some of the things that impact model inference, but we've described them in detail in our paper as well. So model inference we found was impacted by anything that alters biallelic allele frequencies or proportions relative to those expectations. So this could be something as simple as variance or noise in the data. We also found biological processes like mode and timing of whole genome duplication or recent integration impacted model inference. If you also have mapping error or any error, which can be caused by things like divergence from the reference genome, model inference can be impacted. Overall, something to highlight is that model choice in the filtering approach did differ among these data sets. So we highly recommend that you have a sample with sequence data and known ploidy level to decide how you're going to model it and how you're going to filter it to be able to use any of these methods. And because of that caveat, and even though this is the best available approach, I don't recommend relying on this method as pattern-based models are limited by nature. So don't worry, I don't want to do flow cytometry. So we've already gotten started on our next version, NQUAC AI, and we're looking for collaborators. So far, Andrew Pogon and Tripsicum from Alyssa Phillips has been added to our data set, as well as moths from Matt Johnson, and a ton of vaccinium from the UF Blueberry Lab and from Andy Crowell. But if you're interested in joining and you have DNA sequence data with confirmed ploidy level, please reach out. We'd love to include you on our next edition of this method and hope to not do flow cytometry in the future. So to summarize, with AutoPop, we found that mixed cytotype population can persist over time, and we're currently working to extend this to extant populations by adding genotypes, mutation, and migration. With NQUAC, we found the accuracy of pattern-based approaches are limited, and so we're working on a data-based approach that may increase the accuracy. And with that, I want to open it up for any questions and also point you to a link to donate to North Carolina. Galix is part of Western North Carolina, and since it motivates the study, I wanted to make sure to include a link.
All right, well, thank you so much for a wonderful talk. We'll go ahead and, yeah, if folks have questions, maybe feel free to raise your hand and we can go from there. It's probably the easiest place to start. It always takes a couple seconds.
Hi, Jonathan.
Hi, Shelley. Thank you for the very clear talk. Seems to me that, I don't have any idea how you would implement this, but in principle, shouldn't genetic linkage be your friend? Because triploidy is triploidy for the whole chromosome, and that adds information relative to just having individual SNP sites, for example.
Yeah, it does, but it also limits what you can do if you don't have a reference genome with locality. And so we've talked a bit about with our next approach if we should look at, haplotypes or regions or include information about where in the genome each site is. But because of the data that we have, Galix, we put together a genome. It's good for Galix, but it's very disjunct. So when I'm thinking about that data set, I could never use, which chromosome it's on or how is it connected as informative. So I want it to be applicable, but Yeah, that would be great. In an ideal world, I think that would be really informative.
Yeah, a naive question because I'm spoiled by our genetic resources. Yeah,
Yeah,
thank you. Good, great talk.
thanks.
All right, I think Tamsin Dunn has their hand up.
Yeah, hi. Thank you so much for an awesome and really accessible talk. I really appreciate it. I wanted to ask Shelley, are your inputs BAM files? Is it aligned reads?
Yeah.
So why don't you use a variant color, like a somatic variant color that already calls the variants, because then you'd automatically have like all the power of indel realignment and
Yeah.
yeah,
So I thought
all
about
the,
that.
like research that's gone into building variant colors to get your frequencies.
Yeah. So in an ideal world, yes, but because you need to know the ploidy level to call ploidy aware variants, it doesn't end up working. So I've thought about it. I've tried it. It's just not the data. It removes the data that you need. It's a
Did you
step
use like a
before.
somatic variant caller that can call all that's agnostic towards variant frequencies, or were you trying like some?
No. So I should touch a base with you and see what you recommend.
Yeah, get in touch with me because I think at least the option to import VCFs from a somatic variant caller would allow you to harness a lot of rigor that's already been developed, and maybe that'll help you just get those remaining
Yeah,
accuracies up there.
definitely. I'll reach out.
That'd be fun.
A great question. Jeanette. I think, yeah, there you go. Feel free to pop on. There we go.
Hey, Shelley, really cool presentation and also outputs. I was wondering if you could comment on whether or not the on the distribution of errors with respect to ploidy, and in particular whether or not tetraploids are more prone to error, for example, than triploids or diploids, for example.
In the models?
Yeah. In the ploidy inference in NQuack, is it which are the hardest ploidies to infer?
So I wish that was a simple answer because we simulated a lot of data and we had a lot of different data, and it wasn't that it depended on the system. So like in Galex, triploids were super difficult, but only because those triploids probably had recent integration. While for Laria, we actually had issue with our diploids not being called right, which made utterly no sense. And so it has to do with your data and the biological properties it has. And even with the simulated data, it wasn't that one model was better for triploids or for tetraploids or so on and so forth. It was that the data set, how it was simulated really didn't tell you. It's hard to explain, but I went through it for a really long time, and me and Rob Lepore stared at plots to try to understand what patterns we could pull out. And it isn't that these models are actually a good fit for your data. That's the biggest problem, is that there is not a true connection between this math and biology, and so it ends up being very artificial. What's wrong?
Okay.
Yeah.
Okay. Yeah. Thanks.
All right. Well, any other questions before we move on to our second speaker of the morning? All right. Well, thank you so much, Shelley. And if anybody has any other questions for Shelley, feel free to save those to the end and we'll come back around and have a chance to ask questions of both speakers later on. All right. Our next speaker is Michael McKibben. Michael is actually a PhD candidate in my lab studying paleopolyploidy and how ancient whole genome duplications impact plant genomes and plant genome structure. His work today is focused on the frequency of ancient polyploid events across the tree, and in particular, he's working on going to talk about work on a rarely acknowledged challenge, the species tree itself, and how that impacts these inferences. With that, I'll let Michael go ahead and take it from here.
All right, can people see my screen okay?
Yes, looks good.
All right, cool. Thank you for the introduction, Mike, and thank you everyone for attending Mike and mine Shelley's talks today. So I'm going to be talking to you about how we actually infer ancient whole genome duplications on the phylogeny and the importance of considering your species tree when doing so. Polyploidy is pretty ubiquitous in plants, and particularly angiosperms, which from the 1KP analysis, we estimate to have around 3 to 4 ancient whole genome duplications in the history of each lineage. You can see that represented here on the left with every little red branch representing the placement of those whole genome duplications. Now, the ubiquity of these duplications have spurred many interesting questions. Some popular ones I like to think about are how paleopolyploidy impacts the origin of traits. For example, it's hypothesized in the brassicales that paleopolyploidy impacted the emergence of glucosinolates. Whether or not paleopolyploidy is associated with diversification rates in different lineages, and then whether or not whole genome duplications increase in frequency around different geological events, for example, the KPG boundary. Now, across all these analyses, we have some sort of event that occurred in the history of a group of taxa, and we're asking the question whether or not a whole genome duplication coincides with it or is associated with it. And as you can imagine, where we place the whole genome duplication on the phylogeny can impact our interpretations of these hypotheses, and even a simple shift up and down the phylogeny can matter. So how do we actually go about accurately inferring these events and where they belong on the tree? Perhaps one of the most popular methods today looks at the synonymous divergence of paralogs, i.e. gene duplicates in the genome. We typically call these KS plots. The convenience of a KS plot is you only need transcriptomic data, which makes them relatively affordable to do. And on the left in a typical KS plot, we can print it as a histogram. And there's a background rate of small scale duplications that then fit an exponential decay curve as they age. However, as you can imagine in the presence in this right plot of a whole genome duplication, there'll suddenly be a large influx of duplicates that we can see as peaks in the plot. This is all fine and dandy. However, in practice, interpreting KS plots can be a challenge. They can take on all sorts of shapes and characteristics, particularly with fractionation and the nested history of whole genome duplications. And what these peaks actually means in terms of divergence can vary between allo and auto polyploidy. Similarly, interpreting these plots can be a bit more of an art than science. However, we have introduced a tool called Sledge, which is a supervised machine learning approach for classifying whether or not peaks in a plot actually are associated with a whole genome duplication or not. We have a preprint on bioRxiv. I've taken a sample figure. And so on the x-axis, we actually have basically the fraction of the genome that has been duplicated. So 100% would be a whole genome duplication, 50% a triplication. And you can see that we're able to detect them with high accuracy using Sledge and discern from smaller scale duplication events, such as a segmental duplication, where less than 10% of the genome is involved. Now, regardless of what method you choose, once you've found a whole genome duplication, you can try and find when it occurred in a phylogeny using a tool like KS rates. So KS rates uses a series of mixture models fit to the distribution to find our peaks associated with the paleopolyploidies. And then you can compare those peaks to the divergence of orthologs with different outgroups to figure out whether or not those whole genome duplications are shared. KS rates is a particularly powerful approach at doing this because it also allows for rate corrections, given that there's heterogeneity in the synonymous divergence among different taxa. But one problem with this is this is a well-known issue that these mixture models tend to overfit the number of peaks to the distribution. And depending on which outgroups you use during the rate corrections, you may shift the ortholog divergence before or after different peaks. And so when available, if you have a reference genome, people will supplement these approaches with synteny, which is the conserved gene order in different regions of one or more genomes. There are many different ways to visualize synteny. The most popular is dot plots. I have two sampled for you here. On the X and Y axis are different chromosomes either within or between genomes. And then wherever there is gene collinearity between those different chromosomes, you'll see as dots in these dot plots. In the presence of no duplications and relatively few rearrangements, you expect a straight line across. However, on the right here, we see an ancient whole genome duplication that's been fractionated and rearranged, showing up as all sorts of different diagonals in this dot plot. Similar to the KS plot, interpreting dot plots is a little bit more of an art. There aren't too many formal methods available yet for discerning the significance of whether or not these dots are consistent with a whole genome duplication. And so oftentimes we supplement this even further with another visualization approach in the form of syntenic depth. So if I have a diploid outgroup and my species of interest that's a polyploid, even after fractionation, I can count how many collinear loci to the outgroup match to a gene in the ingroup. And so, for example, we have 1, 1. Here, both genes are retained, so that's 2. And we can plot these depths as a histogram. I have an example here in Arabidopsis and Brassica rapa. Oftentimes, they come in pairs. One from the perspective of your outgroup, which, because it doesn't share the whole genome duplication, you would expect to be primarily 1-to-1. In the case of Arabidopsis and Brassica rapa, they do share a much older duplication, so there is a little bit of elevated 2-to-1. But then going the inverse direction, Brassica rapa has a known whole genome triplication, which we can see here as these elevated syntenic depth equal to 1-to-3. The final method, or I should say group of methods for inferring paleopolyploidy, looks at rates of duplication and loss along the phylogeny. And as you can imagine, for example, in O. sativa here, if we see an increased rate of whole genome genome duplication, this may be consistent with a whole genome duplication event. These methods come in a variety of flavors. The majority of them are either gene tree to species tree reconciliations or do some form of subtree counting among the gene tree topologies. To date, the most sophisticated method of doing this is called Whale or ALE. ALE being an amalgamated likelihood estimation. This is a Bayesian approach where you basically represent a cloud of probabilities for different gene tree topologies based off of a given alignment. You can then use them for gene tree to species tree reconciliations. And the benefit of this is allows you to not only estimate rates of whole genome duplications, but also the background rate of small scale duplications, which can have a lot of heterogeneity across your phylogeny. In Whale, there's 2 models you can choose from. There's a critical model, which allows the duplication and loss rates to vary among branches, but equal within, and a biologically more realistic, but also computationally expensive model called the relaxed model, which allows these rates to both vary both among and within. One caveat to Whale is you need some pre-generated WGD hypotheses to place on the tree, so it can operate somewhat akin to a post hoc test. Returning to the 1KP analyses, I wanted to discuss with you the methods that they used here. So this work was done by Dr. Li Zhang on the 1000 transcriptome project, and they used KS plots and mixture modeling for hypothesis generation, and then another tool, which is MAPS, which is one of these duplication loss rate estimation approaches, where you count the percentage of subtrees from the gene trees that share a given duplication on a particular node. You can then compare these to the annulled and positive simulations, which you can see here as these black and gray lines, to discern whether or not, for example, there are 2 peaks in the phylogeny here, consistent with whole genome duplications. Further, they went on, compared their results to syntenic analyses from the literature and found a large degree of congruence. And at the end of all these analyses, they discovered a total of 244 ancient whole genome duplication events, 179 of which were in the angiosperms. Now, one thing studies like this and studies before, I like to think we keep in the back of our mind, but oftentimes don't address, is the species tree itself is a hypothesis. For example, I have 4 hypothetical species here, A, B, C, and D, and 2 potential hypotheses for the relationships among these species. If I infer using one or multiple methods signal of whole genome duplications in species B, C, and D, I might infer very different histories of whole genome duplications between these 2 phylogenies. On the left, there's only a single shared ancient whole genome duplication, where on the right, we have 2. And so that led me to think about how we can begin to address this issue and whether or not the species tree has significant impacts on our inferences. For this, I subsected down the 1KP analysis to 500 species. I'll get into the reason why in a few slides. And I added in 100 annotated reference genomes to do our own synthetic analyses. I then used 2 different dated species trees. One comes from Jansen et al. This is a more traditional approach where you use 2 conserved well-known gene sequences along with 56 fossil calibrations. I then used a service called Time Tree, which you can think of as my alternative approach, which is an interesting tool that averages the topologies and dates across over 2,000 studies from the literature. We then used some of the most recent iterations of the different approaches I've discussed with you so far. For example, KS rates for its powerful synonymous divergent rate corrections. Synteny in the form of MC-ScanX, which also allows us to make these synthetic depth plots we've described. And then Whale to get at this gene tree to species tree reconciliation approach. To give you an overview of the workflow for this analysis, for both the Time Tree and Jansen et al. Phylogenies, we broke them down into 42 subtrees. This is because Whale tends to scale to species trees of only about 20 species. We then generate our initial WGD hypotheses using KS rates, and then test these hypotheses using both of the Whale models along with synthetic depth using MC-ScanX. Now, this is easier said than done. About 600,000 CPU hours later, about 70 years of real time, if you were to run them serially, we finally get some results. I'm going to start with the Janssen's et al. Topology and then zoom in further and further. So this is our merged analyses from the 42 Jansen et al. Sub-analyses. On the phylogeny here, wherever you see a little dot is a WGD hypotheses, and they've been colored by which methods support that particular location. As a reminder, the 1KP analysis has 179 whole genome duplications. And we find that KS rates vastly overinflates the number on this tree. Now, this doesn't necessarily represent 250 unique whole genome duplication events, rather alternative hypotheses for their exact placements based off of what outgroup you use for the rate correction in each sub-analysis. Now, using Whale, we were able to subsect this down to around 170 whole genome duplications, much more in line with what we expected from the 1KP, 32 of which we could further support using synthetic depth inferences. Sort of stepping out and comparing this to how we see in the Time Tree, both methods, both trees show a similar number of whole genome duplications and a similar pattern between methods. So we see that overinflation in both trees from KS rates, followed by around 170 whole genome duplications being supported further by one or more of the Whale models. And this sounds good on first blush. After all, we're seeing a similar number of whole genome duplications and a similar amount of disagreement between methods across both phylogenies. However, this only represents the total number inferred and not their precise placement. Before I give you the total summary of those precise placements, I want to give you a small toy example in the sub-analyses for the Brassicales. On the left, we have the Jansen's et al. Tree. On the right, we have Time Tree. Wherever you see, let me get a pointer. Wherever you see a dark colored 4-letter code, that is a whole genome duplication from the 1KP analysis inferred using KS. We then have in red whenever that was supported further by maps. Underneath, you'll see non-bolded codes representing KS rates inferences from these analyses, and then dots color-coded for whether or not KS rates was also supported by the critical model, the relaxed model, and Synteny. On the left, we have a topology pretty similar to what we understand the relationships of these species to be, and across studies and tools, we largely see an agreement for the placement of these different duplications, with the exception of this duplication in Cobrelinia spinulosa. On the right tree, we see a bit of a different story. I particularly like to emphasize the Arabidopsis beta duplication. This is a well-studied and well-known duplication, which in this analysis, multiple methods supports 2 alternative placements for. This is largely due to Moringa olerifera and Carica papaya being placed shallower in the tree in Time Tree relative to Jansen's tree, which not only causes these 2 alternative placements, but also 2 independent whole genome duplications to be placed by KS rates that we know don't actually exist. Zooming out, to try and compare between the topologies of these two trees, I considered a WGD hypothesis identical if they were at the same MRICA node for identical species in both sub-analyses. And we see, once again, the overall number of whole genome duplications is approximately equal between both trees. However, their precise placement only agrees around 75% of the time. Now, zooming out, what this really means is across both analyses and both trees and methods, we see a relatively equal number of whole genome duplications in all these species that result in around 3.5 whole genome duplications in the history of the average angiosperm taxa. However, the precise placement of those trees does differ based off of the tree topology you use. So if you take anything away from this talk, I hope that you see that the species tree itself is inherently a hypothesis that we are mapping these WGD hypotheses onto, and so they inherit any of the certainty that the species tree might have, regardless of which tool you use to place the duplication on the tree. In the future, I'm interested in understanding how precise branching patterns as opposed to branch lengths influence these inferences. But for now, if you're interested in reading further, you can check out this work in the American Journal of Botany special issue, Polyploidy Twice as Nice. With that, I'd like to thank the members of my lab, particularly my advisor, Dr. Mike Barker, as well as the Department of Ecology and Evolutionary Biology at the University of Arizona. And with that, I will take any questions.
All right, well, thank you, Michael. Yeah, feel free to, as last time, feel free to raise your hand if you have a question, and we've got plenty of time. All right, Doug, go ahead.
Oh, thanks, Mike. Hey, Michael, that was great. It's really good to see this thoughtful dive into the complexity of where we place these and the uncertainty. I mean, I think when we first did this work, we're so happy to be able to make these determinations, but realizing with time, the estimates, may be off and we have to get better, and you're doing that. But, a couple of things. Do you think it's possible, as we use bootstrap values, to give some sort of like confidence level in where a event is placed as one thing? And then the other thing, it's kind of related. When you do these big analyses, you have to remove taxa because they're so computationally intensive. But as we all know, when you remove taxa, that can also have implications for the event goes itself. So does that argue for more focused analyses like a Rosen analysis or a Brassicales analysis or you name it, so that you don't have to sacrifice that important taxon density? And I'll shut up. Thanks.
Yeah, I think those are all really great ideas. I'm particularly hopeful there's methods coming out where they're trying to infer the paleopolyploides and the species trees concurrently. I think that's particularly heavy because, as you can imagine, the duplication itself can also impact our species tree inference. And I think tools like Whale as well also provide us a certain number of power getting at your question related to the probability on different branches. Right now, it's developed where you have a static species tree, and then the gene trees are what are presented in a Bayesian framework. It'd be very interesting to get at where the species tree also has some variability, so we can start to get at trying to not only get estimates of the duplication and loss rates on each branches, but also how confident we are in those rates based off of the confidence in the species tree itself. I'm not sure I fully addressed your question, but those are sort of the first steps I envision into the future of this type of work.
Great, thank you.
Any other questions?
All right, any other questions for either Michael or for Shelley? All right. Well, with that, I'll go ahead and we'll go ahead and wrap up today's webinar. I think I want to thank both Shelley and Michael for being brave to take on the first round of talks and accept those invitations. And it's really great to get this going again. We'll be meeting the first Wednesday of each month. through the new year. And there's a couple emails I got to send to folks. We might add another series at the end. Their schedule's actually filled up pretty quickly. And so there's demand again. That's really great to see. And so our next webinar will be on November 6th, Wednesday, November 6th at 11 a.m. Pacific. It'll be led by Sylvia Canosian and Tamsin Dunn. And so I really look forward to hearing about their research. I look forward to seeing everybody again. We'll see everybody in November. Have a great month. Bye, everyone.
Bye.